Next, let's take a look at inhibition of acid secretion by our parietal cells. In order to do this, let us look through a section of the stomach wall once again. And let's revisit our open enteroendocrine cell, which is the G cell. Recall that the G cell is stimulated by multiple methods, and the purpose of the G cell is to secrete gastrin. The secretion of gastrin by this cell is activated by vagal stimulation in the cephalic phase, and also by amino acids and peptides on their receptors, which trigger the response for secretion of gastrin. Now, the inhibition of gastrin secretion is a classical biochemical feedback mechanism. And I think all of you will be able to understand and appreciate this. And it's also, fortunately, very simple. What I mean by feedback is that an effector cell type must be able to detect changes in the environment. So, for example, the open enteroendocrine cell will recognize amino acids and peptides and respond accordingly, saying that there's food available, so we need to decrease the pH in the stomach, so we're going to secrete gastrin. What this ends up leading to is an increase in protons, ultimately by the parietal cell activity in the lumen. As the protons increase in concentration, they will reach past their binding affinity for receptors on another open type enteroendocrine cell. This cell type is called a D cell. So the D cell has receptors that can bind to protons and recognize the concentration of protons is rising by achieving a threshold of binding affinity. What this then does is stimulate the D cell to secrete the effector somatostatin. Somatostatin is now the hormone that is responsible for inhibition. Somatostatin will enter the blood. It'll also have paracrine effects so that it will be able to stimulate localized environment. And in this case, it will bind to the somatostatin receptor on ECL cells in a paracrine-like fashion. Once it binds to its receptor, somatostatin receptor, now recall that when the ECL cell is stimulated by gastrin, it is secreting histamine, which then is a positive regulator of the parietal cell for acid secretion. Somatostatin binds the ECL cell and now inhibits the secretion of histamine. In an endocrine fashion, the somatostatin also will bind to G cells. Binding to the somatostatin receptor on G cells now will inhibit the secretion of gastrin. Once we have this inhibition all due to somatostatin secretion from D cells sensing high levels, high concentrations of protons, which equates to a low pH in the lumen of the stomach. Now the histamine and gastrin are no longer available. And if we examine what happens to the parietal cell, if this is the lumen, remember we had our antiporter on the surface, our ATPase transporter to pump the protons into the lumen to drop the pH. These resulted from these intracellular vesicles fusing with the plasma membrane of the parietal cell. If we no longer have gastrin signaling through the gastrin receptor, or signaling through the H2 receptor, because these hormones are no longer being secreted, that means that there's no stimulus to push these vesicles to fuse with the plasma membrane. The default pathway is actually to create a new vesicle through the process of invagination of the plasma membrane, and now these antiporters for the proton potassium ATPase pump are being retrieved back into intracellular vesicles. In doing this, this transporter is no longer located in the plasma membrane. It's housed now in a vesicle, and its function is prevented. By doing this, we can no longer pump protons out, and the result is the pH begins to rise.